Hello and welcome to the Malcolm Renfrew Interdisciplinary Colloquium for Tuesday, January 26, 2021. I'm Kenton Bird, the co-coordinator of this series, and with me on the adjoining screen is Dan Buckfitch, the founder of this series and co-coordinator. Before introducing today's speakers, let me tell you a little bit about next week's program as well as a special event on February 4th. One week from today, on Tuesday, February 2nd, we will hear from two University of Idaho sociologists, Kristen Haltner and Dashani Sarathchandra, who will be speaking about their research about climate change skepticism, and they'll be looking at the attitudinal uncertainty among climate change skeptics. So that'll be 12:30 Tuesday at the regular YouTube and Zoom channel. And then Thursday, February 4th, the School of Journalism and Mass Media presents the annual Oppenheimer Media Ethics Symposium featuring David Folkenflik, the media correspondent for NPR, who will be speaking about Trump, the media, and the path forward. And that'll be at 4 o'clock Pacific, 5 o'clock Mountain Time, February 4th. There's a special short URL, pretty easy to remember, uidaho.edu/op21 for Oppenheimer 21. And I'll have more details on that next week. And I'll also be sure that everybody who's on the mailing list for the Renfrew Colloquium receives a special announcement about that program. Today, we'll be hearing from two faculty members in our College of Agricultural and Life Sciences, Shelley McGuire and Mark McGuire. Shelley is a professor of human nutrition and director of the Margaret Ritchie School of Family and Consumer Sciences. Mark is a university distinguished professor in the Department of Animal, Veterinary, and Food Sciences, as well as Associate Dean of Research for the College of Agricultural and Life Sciences. Their talk will describe the opportunities, challenges, and new ways of doing science that the interdisciplinary team encountered as they brought together an international team of researchers, policymakers, and funders. And it will also relate their research to the importance of the university's land grant mission and the university's ability to be at center stage of cutting edge, globally relevant science. And I will stop my share here now so that Shelley can put up her opening slide. And so Shelley will begin the program and then we'll turn it over to Mark at an appropriate point in the presentation. So welcome, Professors McGuire.
Thank you. Thank you very much. So I'm really pleased today to talk on this topic, and thank you for inviting us to do so. It's been quite an interesting few months as we've studied something that we'd never studied before, and that's whether or not the virus SARS-CoV-2 could be transmitted via breastfeeding. So I would like to also propose an alternative title for this talk, and here we go. I really like to think of this last few months as feeling like we've been flying a jumbo jet while assembling it. And that's a phrase that's been used by other disciplines, but this idea of flying a plane while trying to put the wings on and put fuel in it and make sure you've got the right wheels and everything on it. That's how it's felt for the last few months, and we'll talk a little bit more about that. So before I get into anything else, I always think first things first, I'd like to acknowledge everybody that has been part of this project. It's been a massive team effort. And obviously this was led out of the University of Idaho with a lot of really important people that you can see helping build the plane while we're flying it. The project was also involved folks at the University of Rochester, Washington State University, Harvard, University of Washington, University of Arkansas, and Tulane. And very importantly, our funding sources were Bill and Melinda Gates National Science Foundation, and then also some in-kind donations from Medela and Milk Stork. So first things first, big thanks to this group of people. We wouldn't have been able to do anything without every single one of these individuals. Okay, so before getting into this topic, I want to tell you a little bit about this you might think odd McGuire research team. So I'm speaking first, and then my husband is going to speak next. And I want to talk a little bit about who we are, for those of you who don't know us, and why this particular topic was such a perfect fit for what we do. And you'll see here a picture of us at our wedding reception. Back in 1988. We had already been together for 9 years before that. So we're junior high school sweethearts. And you can see in this picture that we actually toasted at our wedding reception with milk, okay? Not champagne, milk, 1988. So we've been working together for a very, very long time, and we've been interested in milk pretty much the whole span of that time. I also want to point out that our team is actually a 3-person team. The third person is Dr. Janet Williams, who is probably the best problem solver that we know. And so this was a really great project for us because it's something that we're very interested in milk for a very long time. And we've also been interested in the microbe in milk. So SARS-CoV-2 is a virus considered a microbe. And so we have a long-standing interest in the topic of microbes in milk. Also, and I'm going to spend a few slides on this, this is a perfect topic for land-grant university, and we have been at land-grant universities our entire careers, and just felt like this was a perfect project to launch into. So before I move on, I want to talk just a little bit about land-grant institutions and why this was such a good fit. As you know, the University of Idaho is a land-grant institution. We have this 3-part mission, which is teaching, research, and outreach. And Mark and I have been very cognizant our entire careers that all 3 of these are important. And for those of you who don't really know the history of how this all occurred, where the land grant system came from, it began quite a while ago, right, with President Abraham Lincoln, who, and I'm trying to get rid of my, trying to figure out how to change something here. Here we go. With the Morrill Act in 1862, the Morrill Act established the first land-grant institutions, and really the basis of that was that U.S. citizens were getting educated in things like Greek and philosophy, but they really felt at that point that we needed practical education that had direct relevance to daily lives. So home economics and animal husbandry were fundamental components of the Morrill Act, and I'm in a home economics unit and my husband's in an animal science unit, so really, really basic parts of land-grant institution. The research part of the mission was established in 1887 with the Hatch Act. This is when the ag experiment stations were established to support useful research. So now we have teaching and research. And to put this into the context of history, a couple years later, University of Idaho was founded. Then Idaho became a state. Then the Idaho Ag Experiment Station was founded, and Mark is the director of that. A few years later, the first college at U of I was established, which was the College of Agriculture. And the next year, the Department of Domestic Sciences, later named Home Economics and now Family and Consumer Sciences, was established. And in fact, our unit is the oldest standalone department at the University of Idaho. So the research part of the land grant mission was established in 1914. This established the Cooperative Extension Unit, and that was to disseminate new information to the public. So that's the outreach component. So really this whole land grant mission, both of the schools that we're in, I'm in the Margaret Ritchie School of Family and Consumer Sciences, used to be called Home Ec, and Mark's department, Animal Vet and Food Sciences, are really 2 of U of I's quintessential land grant units and really fit the project that we're going to talk about today perfectly. So just another couple slides about this and the School of Family and Consumer Sciences. I think a lot of people at the University of Idaho don't really understand what we do in the School of Family and Consumer Sciences, and why somebody like me, who's a hardcore nutrition physiologist, is in the school. And I'll just A little bit of history. We are the university's oldest department. We actually started having classes in the late 1800s. Here's a picture of some of our first classes in 1895 and our first graduating class in 1897. The unit was then established in 1902. So the classic domestic sciences disciplines are things like food safety, nutrition, which is where I come from, clothing, housing, child development, family studies, and personal and family finances. So most nutrition programs in the country actually were part of an initial domestic sciences unit. But we've come a long way since then. You can see here pictures of the 1897 class and what was going on at the time, and now our current Family and Consumer Sciences, which is not your mother's home ec class. So these domestic sciences departments were actually they came out of the women's rights movements and the suffragette movements, and very, very closely associated with that, with the idea that women needed to go to college and that we could apply scientific principles to everyday activities, and in fact, to give women more time to do what they needed to do in the world around them. And I will say that I believe that the founders of the Margaret Ritchie School of Family and Consumer Sciences would vigorously approve of our COVID-19 work. So our unit was renamed to the Department of Home Ec in 1911. We moved out of the College of Letters and Sciences to the College of Ag, now CALS, in 1982. And we were renamed the Margaret Ritchie School of Family and Consumer Sciences in 1993. So in fact, we're central to the land grant mission. I am a human nutritional physiologist housed in a classic FCS unit. And in all of my work, I teach, research, and inform the public about optimal nutrition. And my specialty is around moms and babies and breastfeeding. Similarly, Mark is a dairy scientist nutritionist housed in a traditional animal science unit, again, with this three-part important outreach, teaching, and research activities. And of course, his is focused on the dairy industry and the importance of dairy foods to human health. So again, just establishing the teaching, research, and outreach components of the land grant mission. Okay, so with that as a little background and to understand how we fit into the whole land grant system and how this is very pertinent to this topic, we'll get back to the topic of today's talk. And that is that when the pandemic hit, there truly was a global crisis, because at the time we did not know if this virus could be transmitted from mother to baby via breastfeeding. And we know of several other pathogens that can be transmitted. For example, HIV is well known to be transmitted from mom to baby during breastfeeding. We had no information on this at the beginning of the pandemic, and we had almost no information on any of the coronaviruses. So when the pandemic hit, this caused infected moms to be separated from their babies around the world. And that is not happening so much anymore, partly because of our research, but you can imagine that this was a global crisis immediately at the beginning of the pandemic. And one thing that we saw also at the time is that the first few papers that were coming out on the topic, we could not trust. They were very poorly conducted. And a lot of this is because human milk is actually very difficult to study. You really need to validate your assays that you're using. You have to collect the milk appropriately. You certainly don't want to contaminate it. And so we saw a need immediately for doing studies, doing them well, and being able to then disseminate what we were finding to the public so that we would know how to move forward with this. Again, a perfect land grant challenge. It's relevant to the general population. Absolutely needed vigorous science. We had to get it right. We were impacting the lives of a lot of families around the world. And again, had both local and global impact. Anywhere where moms were having babies or moms were breastfeeding babies needed this information. So to give you a sense of how doing science on a pandemic during a pandemic looked, I just put together a really abbreviated timeline. As you all know, January, February, totally normal. And then of course, in March, life as we knew it came to a screeching halt as we were all sent home to try to work from home and not spread the virus. And I call this crisis 1, which was the beginning of the pandemic, and we'll talk a little bit about crisis 2 at the end of the talk. So everything went into fast forward, just quick, fast gears at the beginning of this when we were sent home, and we realized that there was this public and global crisis around breastfeeding and whether moms and babies should be separated, which you could imagine is a horrible situation. And I put some actual dates on here so you could get this sense of how quickly we moved. March 2020, our group actually coalesced a group of national experts via phone calls and Zoom, breastfeeding experts, milk experts. And we talked about the topic. We realized it was going to be a major issue. We put together a Slack group, which is one of these new platforms, you know, where you can all communicate. It was new to us, but we had a postdoc who knew how to put that together and get it going, run pace. And so the next day we put that group together. We now have over 100 researchers on that Slack group, and it was really integral for us to Maze. We had actually collaborated on an effort to establish milk collection guidelines for collecting milk during this pandemic, because the studies that were coming out, like I said, were not well done. And we wanted to make sure that the researchers doing this sort of work were doing it well. And we published those to our society's website. We also started summarizing breastfeeding guidance that was coming out around the world, a lot of it saying that moms and babies should be separated. We began compiling a literature review. And importantly, we started sharing documents like IRB documents. So this is the Institutional Review Board. Every time you do a human subject study, you have to have all of your procedures approved by the IRB. This is a process that often takes many, many months. And to help speed the process up, we started sharing documents with each other, sharing instructions, sharing protocols, and we were doing all of this via Slack and Zoom. Literally within about 2 weeks, we all started submitting IRB applications to our home institutions, trying to get ahead of the game because we knew somebody was going to have to study this and they were going to have to study it well. Within another week, we actually submitted a literature review on the topic for publication. And on the bottom here, I've put sort of the state of the science when we started writing this paper and then when it was published a month later. When we submitted the paper, when we started writing the paper, there were 9 publications on the topic. 16 women had been studied, 20 milk samples had been studied. All of them were negative for the absolutely no information in any of these papers on how the samples were analyzed. And in my book, that just validates the studies. McGuire was published. There were 25 publications with over 40 women. You can imagine, just ramped up the whole issue even further. But again, almost no information on sample collection analysis. But this paper was published in April, and it's already received, I believe, over 70 citations from other researchers. It's also way out of date now, but people are still did a Gates grant. Literally, we wrote it in a weekend and it was funded the next week. By the end of the month, we had submitted an NSF grant application and it was funded also within a week. We started collecting milk samples for both the Gates and the NSF studies in May and June. Sometime in June, we started writing up a formal milk sample collection protocol. That was actually key to being able to do these studies right. And we'll come back to this, but by September, we were actually publishing preprints or basically non-peer-reviewed results from our first study, which I have to say didn't feel so great. As classic scientists, we typically like to wait till we have peer review in order to put out our results, but in this new world, the new thing is goes to peer review. Fast forward a little bit, in December, crisis 2 happened because that's when the vaccines came out. Mark's going to talk a little bit about that. And here we are in January when our formal milk sample collection protocol has been published, and our first peer-reviewed Gates results should be published any day now. So that's kind of what it looked like. And to put this in another perspective, I put together a bit of a scientific process we normally use for human subjects research, and it usually goes something like this. We ask a question, okay, then we think about the best study design to answer that question. We make it accurately. It usually takes months, and it takes more months to get them funded. Then if they're funded, we hire personnel. Then we do the IRB approval. We get biosafety approval for everything that we're doing. We conduct the study, we do lab analyses, and it goes through peer review, and then it gets published. And we do public outreach so that we can disseminate our results. This is the normal process. Each one of these stages can take, 12 to 18 months, maybe 6 months, another 2 years for that, and another maybe 10 months for that. So typically, a study of the type I'm going to describe today would take anywhere from 2 to 3 years or more to happen. This is not business as usual. You just saw how all those arrows moved around, and this is the actual order that we did these things in. So we asked the question, first step, fine. Then we got IRB approval. Then we started making sure we had the right personnel. Then we obtained funding. Then we got our approvals and designed the study and validated the methods and conducted the study. We did the lab analyses. Then we did public outreach. And now finally we're going through peer review. And so everything was out of order. And again, it felt like this flying a jumbo jet while assembling it. And this whole process has taken about 10 months rather than 2 or 3 years. Some of the issues that we have encountered, of course, the first is, as I just mentioned, we needed to do the research in very out of order sequence. That's incredibly risky for a scientist and didn't feel very comfortable most of the time. Most of us were working from home, some exclusively from home, some with small children. And I will say that the McGuire-Williams research team was incredibly dedicated and world-class to get all of this done. None of this was easy. In addition, we're studying a serious pathogen, right? And we needed the same chemicals and assay kits and PPE needed by everybody else and in very short supply. So we literally begged, borrowed, we didn't steal, but we begged or borrowed some of this stuff from colleagues. It's been difficult to get. We didn't know how to collect, ship, receive, analyze the samples as they contain pathogens. So the lab had to figure that out. Janet and Ryan had to figure that out. We needed IRB and IBC approvals. So these are the safety approvals before we could recruit subjects. That can often take many, many months. And I will say that U of I's Institutional Review Board and Institutional Biosafety Committees were absolutely amazing in helping us get this done. We also needed COVID-19 infected women. Obviously, we didn't have many in Moscow, Pullman in the first months, and thank goodness for social media to find them. And of course, we needed money. These studies are expensive. And it turns out there was almost no emergency funding available from normal sources. So for example, we are funded currently by the NIH, National Institutes of Health, and it typically takes 1 to 2 years to get money from the NIH. They had absolutely no emergency funding, just shocking to me. We did initially get, we got early money from the Gates Foundation, and we do have a saying in our lab that when the Gates Foundation asks you for something, you give it to them. And they literally reached out on a Friday, asked us to write a grant application. We had it in, I think, on Sunday, and discussed it with them the following week, and they funded it. So thank goodness for the Gates Foundation. And I will just thank the UI and CALS grants offices for helping us get these grants out the door in record time. It was really amazing. So I'll tell you a little bit about what we did for the study, and Mark's going to tell you about what we found. Our hypotheses from the start were that in lactating women who were recently diagnosed with COVID-19, that we would find some virus in their milk, not all milk, but we expected to find virus in some milk samples. We also were very interested in whether the virus was in the milk or could be obtained by the infant via the process of breastfeeding. So we actually swabbed the breasts before and after cleaning to see if we could detect the virus, and we hypothesized that we would find the virus sometimes on the breast prior to but not after cleaning. We believe that milk would contain antibodies that neutralize the virus and that the antibodies in the milk would be long lasting. To do this, we used the following study design. We used what's called a prospective longitudinal repeated measure study. So that means prospective, you're looking at things, you're evaluating things while they're happening. Longitudinal meant we took repeated sample, or we were looking at things over time, and we took repeated samples from each woman. So in the end, we studied around 50 moms and babies. The moms were at least 18 years old. Of course, they were lactating, breastfeeding, and they had to be recently diagnosed with COVID-19. And we were looking for women who had been diagnosed within the last week, because we wanted to make sure that they were truly infected and recently infected. We collected milk and breast swabs using very, very standardized protocols, which was incredibly important because we wanted to make sure that this was done in the best way possible. We collected these samples 1 to 3 times in the first week following diagnosis, and then again monthly-ish for the next 2 months. We collected breast swabs again before and after cleaning, and those were collected before the milk was collected. We analyzed the milk samples and the breast swab samples for the virus, and actually we were looking for the genetic material or the RNA in the virus, so that's what we analyzed. And just to also tell you, we analyzed 10 pre-pandemic milk samples so that we would have some control milk. We also analyzed the milk for antibodies specific to the virus, and then the ability of the milk to inhibit or neutralize the virus. And just to give you a sense as to how this looked and how we did this, and again, to have a call out to the McGuire-Williams research team, because they really just made this happen. We recruited via national Facebook ads. And then when a woman contacted us from anywhere in the country, they were interviewed, they were consented if they could be in the study, and then they were helped by our study personnel who were working from home. So this is just a picture of a home office. This was all done mostly from home in people's bedrooms and makeshift offices. Then we had, as I mentioned, we had the milk and swab collection kits were very, very standardized. They were made here in the Nichols building. This shows Bethany Fahrenkamp putting together these kits in full PPE. The kits were shipped overnight. This is Alex Gogol, a grad student, shipped them overnight to the women. These kits were received at their homes. The women then collected the samples and did the surveys at home, and then we used a company called Milk Stork that picked up the milk and shipped it back to the University of Idaho, where they were very carefully handled and then analyzed. And yes, this was all done while U of I and Moscow had stay-home orders. With that, I'm going to pass it off to Mark to tell you about the first publication.
So the first publication will be out momentarily. It's fully approved by MBios, so it'll be online. We were hoping today, but in the next day or two. And in that publication, there's a lot of details with regards to the validation of the assay, which I think was a critical component of the work. Ryan Pace and Janet Williams extensively analyzed the assay to make sure that it would be valid for human milk, meaning that they spiked samples with inactivated virus and were able to detect an additional spike. We put milk in refrigeration versus -80 because we knew we were shipping these samples across the US that they might be thawed or they might come warm, and we wanted to know what the stability of the virus was, so that at least the viral RNA, so that we could confirm that we were actually measuring something that was real. And so tremendous amount of work in the validation steps that occurred in May and June, and using the assay very similar to what's used to test the U of I students and staff and faculty. It's the CDC assay. With regards to the subjects that were enrolled, this is from the first paper where we have 18 subjects in the first week after diagnosis. You can see that the women were 34 years of age, a variety of races and ethnicities, but mostly white, non-Hispanic. I don't mean to be crude here, but they're a little bit heavy, large body mass index. And then you can see that we're dealing with women that were about 6 to 7 months postpartum. With regards to symptoms, there were 3 that were asymptomatic out of the 18. Most had symptoms beforehand, and we'll describe those symptoms shortly. Of the infants, half were female, and you can see the fairly normal in birth weight, birth length, and most of them were at a stage of breastfeeding where it was mixed feeding and not exclusively breastfed. A few of the infants were tested for COVID-19, and 6 were positive for that test. This figure shows some of the symptoms that the patients had. Of the 18, you can see the most common ones were those that you read about all the time: the loss of smell and taste, and headache, fatigue, diarrhea. I think we would classify that most of the subjects had relatively mild symptoms, not very severe. And here's the sampling that occurred relative to the day of diagnosis. So if you look at the figure, time 0 is the day of diagnosis, but you can see that some of these subjects did test positive well before that time that we called time 0. Some of those subjects had multiple, if you look at Q&R, had multiple positive tests over the time. We tried to relate the onset of symptoms as well, and so those are the red triangles. And then the milk collections occurred in those yellow circles. So you can see that we have a number of samples within the first week of diagnosis, and this really is, I think, a hallmark of the work, where many of the other studies have a few samples in a period of time around diagnosis and disease, but many actually had samples in previous publications, had samples either before or well after the diagnosis occurred. So I think this really is looking at the highest risk period potentially for transmission. Again, we were looking for the genetic marker for SARS-CoV-2 in milk using that real-time PCR assay. And main result is pretty easy to say. Of all those milk samples that have been analyzed in the lab, none have contained any detectable SARS-CoV-2 RNA. And now Janet and Ryan and the team have expanded that to nearly 50 women in nearly 300 milk samples. But what's interesting is there was breast swabs collected before and after washing of the breast, which occurred before then the milk sampling. And there is evidence of the SARS-CoV-2 RNA present on those swabs before the cleaning. And this could be a reason why some of the previous publications actually detect the RNA in milk. It could simply be that that is being picked up as the milk sampling occurs off of the skin. And in general, those samples taken before breast cleaning are 15% of those samples do have detectable RNA. One of the major findings for this work compared to previous work is that there are antibodies to the SARS-CoV-2 virus. If you look at the figure on the right there, we have 3 targets. So those are molecular targets for the receptor binding domain and the spike protein and the nucleocapsid protein of the SARS-CoV-2 virus genome. And you can also see that there are two common cold coronavirus targets as well in that figure. The box plots have the samples from the infected women are shown with the red circles, and those from the pre-pandemic controls are shown in the blue circles. And so you have two boxes here where you're comparing the two, where you're comparing a single target, molecular target to, well, protein in this case, for antibodies against it. And in the case of what we found is that there was a significant difference in those subjects who were infected in antibodies to the receptor binding domain, not to the spike protein, not to the nucleocaspid. It appears that there is some common thread to those antibodies having history, and you can see that those antibodies are also present to those common cold viruses. So there's a knowledge, if you will, in these women's immune system to coronaviruses, but the specificity to SARS-CoV-2 is really tied to that receptor binding domain. And what's mostly incredible is that these antibodies appear in milk very quickly after infection. And so time zero here again is day of diagnosis, and you can see that the antibody to the receptor binding domain increases rapidly and reaches a plateau in milk approximately day 18 after that diagnosis. And what's nice about these data is that these are, again, longitudinal data on women, and you can see that this response is extended through, in this case, about 2 months after diagnosis. And so there is a long-lasting impact of potential protection to the infant from these antibodies in the milk. Of course, the antibodies don't necessarily tell the whole story if they don't do anything. And our collaborators at the University of Rochester actually tested whether or not our milk samples neutralized, effectively, if you will, killed the virus and its ability to infect cells. And what they found is that these antibodies, particularly to the receptor binding domain, were correlated very well with the ability to inhibit the growth of SARS-CoV-2 in culture. And so these antibodies are present, and these antibodies are effective at potentially fighting off an infection. So quick summary here, there's no evidence of the SARS-CoV-2 virus in milk produced by infected women. Could be found on breast swabs collected. We do detect it on some breast swabs collected before washing. There are antibodies present in that milk from infected women. And most of the milk from those infected women did have neutralizing capacity against the virus, while the pre-pandemic samples did not. And that really suggested that antibody to the receptor binding domain is very important in this story. Of course, we didn't evaluate a whole slew of different antibodies that could be present, but in this case, that receptor binding domain really stood out. And that neutralization was correlated with the antibody concentrations that were found. And luckily, it appears that those antibody concentrations stick around for a while during lactation and can be transferred then to the infant. So overall, our data provide evidence that breastfeeding, at least in the case of mild to moderate infection, does not pose a risk, and it likely provides long-term benefits to the infant. So I wish that was the end of the story, but there is a substantial amount that still remains to be studied, and our data do not represent all the cases that occur during lactation or breastfeeding. We certainly have a couple of samples from women with severe disease that we have yet to analyze. We hope to do so soon. We don't know whether stage of lactation, early lactation, late lactation has an impact, or whether degree of nutritional status, particularly that might be occurring in third world countries, how that could impact transmissibility of the virus in milk. One of the hallmarks of HIV transmission through breastfeeding is the presence of mammary inflammation. We did assess inflammation in these subjects, but again, we don't have maybe sufficient range of that inflammation to know whether or not it's not impacting potential transmission or presence of the virus. We have to make sure that the scientific community looks at these other populations to ensure that the message is clear that breastfeeding is safe. We don't know whether that viral RNA that we detect on the breast skin represents viable virus. Sadly, the samples only contain enough to do the RNA assessment, the real-time PCR assessment, and so we don't know whether or not this is a sufficient risk to the infant in transmission. Certainly something that should be studied in the future. And we also are examining whether or not this infection has any impact on milk composition or other milk components that might have long-term either positive or negative effects on infant growth and well-being. So Shelley suggested there is a crisis too that we're now re-engaged in. We thought that the COVID work was mostly over, but sadly there's another issue that has arisen, and that is the question of whether breastfeeding women should be offered the COVID-19 vaccine. And the reason there is a question mark there is because simply pregnant and breastfeeding women were not included in these vaccine efficacy and safety trials to really know whether or not it was either safe for the woman or safe for their infants. And so there are questions that need to be considered here. Do components of the vaccine get into milk? Does it affect the milk composition? And ultimately, are there effects on the infant? In some regions of the world, the governments have stated pretty clearly that women either must choose to get the vaccine or stop breastfeeding and stop breastfeeding, or they can continue to breastfeed but not get the vaccine. The vaccine is recommended for breastfeeding women by the CDC, the World Health Organization, but there are certainly concerns and imprudent concerns by women around the world. So at this point, with regards to the second crisis, we are currently working with colleagues in New York City at Mount Sinai on the Pacific Northwest National Laboratory to answer the urgent questions of how do these, do we see any components of the vaccine in milk, and does it affect milk composition? So we certainly have a new study, and we have lots of interested parties and willing subjects, as many of the health professionals are women in the reproductive years. At this time, we were looking for funding to support these analyses. So how can we be better prepared for the next pandemic? Shelley has in particular been involved in groups associated with the World Health Organization, CDC, and NIH to put together documents that hopefully will be found in the literature in the future that will really identify the best practices so that future pandemics can be studied very quickly with care, and so that the correct answers are obtained. We should strongly urge public health agencies to consider breastfeeding transmission and vaccination of breastfeeding women when they think about this in a pandemic right from the start, not after the fact. And one of the issues, as Shelley mentioned, is that milk has a number of complicating factors when it comes to analyses of any component. There are RNases, proteases, DNases present in milk, and they all potentially could impact any laboratory analysis giving false results. And so make sure that all assays are validated for milk. And we need to have that emergency funding opportunity to really study this in the future. So The last slide here, just returning to our land-grant mission. That mission is really providing the teaching, research, and outreach to impact everyday people. And it provides the intellectual capital, provides the research infrastructure, and it provides the extension or outreach component such that we can rapidly respond to any emergency that happens around us. So I think because of our presence in a land-grant institution, we're substantially more nimble than most of our colleagues. And that goes to really the infrastructure throughout the entire university. And so while it's been a lot of labor by a lot of people, I don't believe that our research would have been anywhere near as successful as without the components that occur within the university. I'll just put it bluntly. I think there were partners that we had at the beginning that still have not received IRB approval, and yet we were able to obtain it right from the get-go. So thanks for your time, and I think we're available to answer any questions. Again, it was a huge team effort.
Thank you, Professor McGuire and Professor McGuire, for the timely and important presentation today. And I would like to invite our audience of both scientists and non-scientists to submit a question in chat if you're so inclined, or if you're bold enough to just unmute yourself and ask the question verbally, we can do that as well. Maybe just to get things started, I'll throw one out. So what are some of the misconceptions about COVID-19 and mother to child transmission that you've had to overcome? Are there some stereotypes or misunderstandings or myths out there that where you've needed to set the record straight?
Well, I think to have a myth, you have to know the truth, right? So from the start, we didn't know what was a myth and what wasn't. So I will say that there's a whole group of sort of breastfeeding advocates that never want to consider the possibility that there might be a downside to breastfeeding. Right. So from the beginning, we didn't know the answer. We thought it was possible. We thought it was possible that this was a mode of transmission, and we wanted to stay objective as possible. And I did find myself sort of cringing early on by these breastfeeding advocate groups that were just saying, it's got to be good. It's got to be good. It can't be bad. And I kept thinking, well, what if it is? So I don't know if that's a myth, but it was something we had to deal with from the very start.
We've got some great questions and observations in the chat, and maybe I'll start with Nick's. Have there been any tests with other mammals that yielded different results?
I'm not aware of any SARS-CoV-2 work in other animals at this time, but a previous coronavirus, MERS, the Middle Eastern respiratory virus, coronavirus, was fairly well studied because it was present in camel milk.
Although it may have been contamination.
But it could have been contamination from the subjects. It is not clear that those animals were sampled well enough to preclude that it couldn't have been on the skin or elsewhere.
We did try to collaborate with a group that was studying bats as an animal model. And we were going to try to milk bats, but that hasn't come to fruition yet.
Connor has a couple of good questions. He wonders if antibody neutralized virus was found in breast milk, would this imply that women who have not yet developed antibodies could still have virus in their milk?
Great question, Connor. So not all of our subjects who were infected had antibodies, and not all the milk samples neutralized the virus. But your point is also to add to your point, we do have samples from non-infected women that were also looking for the presence of antibodies to suggest that there could be a relationship. But I guess my first point is not all of those women infected had antibodies or neutralization.
And Connor's follow-up is, have other vaccines in the past ever caused issues for breastfeeding women or their babies? So a vaccine for polio or flu or pneumonia, any of those?
You know, that's not my area of expertise, but nothing's coming to mind. I mean, there are more problems in pregnancy, so there's concerns there. And in fact, there is some concern about the current vaccines in pregnancy, but not so much for breastfeeding. If there have been, they've been minor and rare.
Betty asks, why were some babies tested and others not? And do many babies get sick by just being near an infected mother, I guess, through conventional transmission methods for viruses?
Yeah, Betty, good question. So, you know, we did a lot of this work at the very beginning of the pandemic, and the testing, you know, there weren't that many tests, right? So babies weren't, just in general, they were not getting tested. Also, babies, you know, we're lucking out with this pandemic. Babies don't seem to be getting very ill. I mean, obviously there are some cases where they do, but these babies with that are positive tend not to get that ill. So I think the answer is a combination of things. The babies just tend not to get tested, and they weren't that ill.
The second part is, do babies get sick just being around infected moms? That is a part of the NSF grant that we received that we're still studying. And so we had a contribution from some anthropologists who deal with maternal infant care. And when our study personnel talked to these subjects at 3 times in the first week, week 2, week 3, week 4, week 2 months, there was a long survey that talked about who was with the baby. Did anybody else in the family come down with COVID? What are those interactions? And so we're just starting to look at those data and try to tease out whether or not there was any transmission. And in that case, we also have, I lost my train of thought there. In a couple of our subjects, actually, the moms were separated from the babies. So that it's an interesting comparison what we have there. So they were pumping milk and dads were feeding it. And so interesting experience.
Yeah, you know, we now know that so much of the transmission is due to families, right? Living situations. And to be honest, it's next to impossible in something like this to separate out, you know, is the baby getting COVID from the milk? Is the baby getting COVID from being around the mom? I think, you know, there will be enough data in the next several months to be able to answer those questions, but we don't have the answers yet.
So Ramsey, would you like to turn on your camera and unmute yourself and make a comment or ask a question aloud?
Yeah, so I had a question for you guys. That's more of an anecdote. So my kiddo and we actually both got COVID around the same time, except my baby, who was 10 months at the time, he showed his symptoms about 2 days before I did. And we continued to breastfeed the entire time, and we didn't get diagnosed till like a few days after. So I was just kind of curious, because I've heard that it's the mom that passes it to the baby, not the other way around. And I've also heard that when a kiddo gets sick and they breastfeed, the mom incorporates the virus and is able to produce the antibodies for the baby who can't produce them. I don't know if you're kind of understanding what I'm trying to get at, I guess, but I don't know. I just thought it was a really interesting situation, and I was really excited to hear what you guys had to say about breast milk and antibodies and stuff. So.
Yeah, there's a lot of layers in there, and I'm sure I'm going to forget some of them. Can the baby pass it to the mom? Absolutely. I mean, absolutely, right? Any infected human can pass it to somebody else. Interestingly, it might be possible that if the baby has the virus and the virus is in the baby's mouth, we now know that anything in the baby's mouth actually goes into the breast at every suckling. So I don't know if that would cause infection in the mom, and this hasn't been studied, so I'm just philosophizing here. But we do have evidence from other studies and other work, pre-COVID, that when a baby is ill, that the milk that she produces actually has more immune cells in it. I don't know if we have data that she produces antibodies toward what the baby has, but there is growing evidence that mom and baby are sort of a single unit, right? And when the baby gets sick, the mom responds. And when the mom gets sick, the baby responds, which is a beautiful system if that's really what happens. And I would like to believe that that's true. And I think it probably is. We don't have any good data on what you're talking about, like, transmission from an infant to a mother. Those things will come out in the next months, as we get larger data sets. What did I miss?
I think you got it. Yeah, no, that was great. Thank you.
I'm glad you look like you recovered well, so that's good.
Yeah.
So a question from Jeremy, can breastfeeding confer some passive immunity to the baby through the transmission of antibodies? Either one of you want to tackle that?
Oh yeah, I mean, we believe so, absolutely. And we're also very interested in, of course, in whether maternal vaccination is going to do the same thing. And we're interested in whether that passive immunity is the same if the mom actually got SARS-CoV-2 versus she got the vaccination. But I think there's no reason to think that the baby would not get passive immunity in that way.
And Jagdish has a more technical question, and I hope I get this right. Did you by chance check the antibody neutralization against RBD from the newer SARS-CoV-2 strains?
We tested it against one strain, so no. The answer is we are not.
The Hong Kong strain.
Yeah, the Hong Kong strain. We have not looked. Our collaborators have not looked at the new variants that are out there. The multitude of new variants. I'm hoping that those antibodies don't have that level of specificity. But I am concerned because it appears that any change in that RBD structure does affect the ability of the virus to bind to the host receptor, and so it's worrisome. But I'm hoping that that antibody is not quite that specific.
Thank you.
So you had mentioned that you thought the University of Idaho was more nimble, perhaps, than other land-grant institutions, and having taught at two larger land-grant institutions before coming to U of I can attest to that. But could you talk a little bit more about why that is? What were you able to do differently here that you might not have been able to do somewhere else? Or where did the efficiencies come in, say, seeking the approvals from IRB?
So I'll speak to that first. I've been at the University of Illinois, Cornell University, and then Washington State University for 23 years. And every time we had to get approvals for everything, it took months and months and months. Even at U of I, before the pandemic, it never took months and months and months. So we started from a more effective baseline. And then I think the interpersonal, just the interactions that we have here at the U of I are so much friendlier. There's so much more relational. I'll give you an example. When we first reached out to the IRB, and I just said, I think we're going to do this. I don't know who we're going to do it with. I don't know who's going to fund it. I don't exactly know what we're going to do, but we have to get the ball rolling. And Jennifer Nealon over at the IRB, she actually set up a Zoom meeting with herself, with the biosafety folks, with legal, you know, with like everybody that was going to have to approve this. And within, I think, 24 hours, we were all talking together. And they threw up no roadblocks. Everybody just jumped in, worked together. And I've never seen that at another university. Amazing.
I think, and I see Megan, the biosafety officer, is on here, and a lot of kudos to Megan, who, you know, she volunteered, came in the lab, went through to make sure we had the procedures in place so that the biosafety committee would approve that we were handling this milk in a safe manner. It was just that everybody stepped up. They didn't hide because of COVID. They did their jobs, and they did their jobs well. And it was just a big team, a real big team, and everybody pulled their weight. And it was a terrific experience, although exhausting.
Well, that's great to hear. Another example of the Vandal family coming together to support an important research effort. I think we've got time for one more question, and Ray, looking forward, is asking, are you looking for breastfeeding volunteers to continue the study, particularly the effect of the vaccine on breast milk?
So we're working with colleague Rebecca Powell at Mount Sinai, and she has more milk. She has literally thousands of women volunteering to provide milk. So we don't need milk on that front. However, having said that, we do have two studies going on right now. If you're a breastfeeding woman and would like to participate, you can send me an email at smcguire@uidaho.edu. We'd love to talk to you.
Well, that's great. Thank you so much for this informative presentation. And for our audience, if you are not receiving our weekly announcements, I put the link, just class MRIC email announcements will let you sign up. And this program has been recorded and will appear on our YouTube channel probably later today or first thing tomorrow morning. And it's also on the Colloquium Archives page. So hope that you will, if you have friends who missed today's program who might like to see it later, there's an opportunity to do that. Before we adjourn, I'd like to invite everyone to turn on your cameras and give a virtual round of applause, or in Zoom if you want to put your two hands together for applause as well. This has been terrific. Thank you for your time, and keep up the good work. We may want to check in with you in the fall and see what new results you have uncovered by then. I hope you can join us next week when two of our sociologists will be speaking about climate change skepticism, 12:30 Tuesday on February 2nd. So looks like the sun is shining in Moscow. Hope you can get out and enjoy a little fresh air this afternoon. Have a great day. Thanks for attending, and stay well.